The science of dying. The science of dying explores the intricate biological processes that occur as life nears its end, the stages of death from a medical and cellular perspective, and modern advancements that are extending life and understanding the aging process. This area of study combines knowledge from medicine, biology, and technology, aiming to uncover both the causes of death and the potential for delaying it. Biological process of dying. As a person nears the end of life, the body undergoes a series of physiological changes in response to the failure of organs and systems. While the exact sequence of these changes varies, several common biological processes signal that the body is shutting down. Decreased blood flow and organ failure. Blood circulation slows down as the heart weakens, leading to reduced oxygen and nutrient delivery to vital organs. This is especially critical in the brain, where a lack of oxygen results in rapid decline in mental awareness and consciousness. Changes in respiration. Breathing patterns change significantly in the dying process. Cheyne-Stokes respiration, characterized by cycles of shallow breathing followed by periods of apnea. Temporary cessation of breathing is common and signals a decline in brainstem activity. Metabolic imbalance. As organs start to fail, the body's metabolic processes become irregular. Waste products accumulate, leading to toxic buildup in the bloodstream, which exacerbates organ failure and hastens death. Loss of reflexes and muscle control. In the final stages, voluntary and involuntary muscle functions diminish. This includes the loss of reflexes such as swallowing, blinking, and eventually, the body's ability to maintain a heartbeat. The biological process of dying is complex and varies depending on the cause, for example, disease, age-related decline, or trauma, but ultimately, it leads to the irreversible cessation of bodily functions. Stages of death. Clinical, biological, and cellular. Death can be understood in stages that reflect different levels of bodily shutdown, with clinical death often preceding biological and cellular death. 1. Clinical death. Clinical death is defined as the absence of a heartbeat and respiration. In this stage, the body can no longer pump blood or oxygenate tissues. Clinical death may still be reversible for a short period through resuscitation techniques such as CPR or defibrillation. During this time, brain cells can survive for a few minutes without oxygen, but prolonged oxygen deprivation leads to irreversible damage. 2. Biological death. Biological death occurs when vital organs, particularly the brain, suffer irreversible damage due to lack of oxygen and blood flow. This is considered the point of no return, beyond which resuscitation is impossible. Brain death, a subcategory of biological death, is used in many legal and medical contexts to determine death. It signifies the complete and irreversible loss of brain function, including reflexes and responses, marking the individual as legally and medically dead, even if other body parts may still function temporarily. 3. Cellular death. Cellular death occurs as cells progressively die after the organism has expired. Different types of cells die at varying rates. For example, brain cells die within minutes, while skin cells and bone cells may remain viable for hours. Cellular death is associated with autolysis, or self-digestion, where enzymes break down cell components, eventually leading to tissue decomposition. This process is influenced by temperature, the body's environment, and internal conditions. Each of these stages provides insights into how the body transitions from a living to a non-living state, guiding medical professionals in determining the appropriate time to cease resuscitation efforts and enabling scientists to study cellular processes for potential anti-aging applications. Advances in life extension and aging research. As our understanding of aging and death improves, scientific efforts to extend life and slow the aging process have made significant strides. These efforts span a variety of fields, including genetics, regenerative medicine, and pharmacology. 1. Genetic research on aging. Telomeres and aging. Telomeres are protective caps at the ends of chromosomes that shorten with each cell division. When telomeres become too short, cells enter a state known as senescence and eventually die. Researchers are exploring ways to extend telomeres or delay their shortening, potentially slowing the aging process. SIRT genes and longevity. The certain family of genes, SIRT1, SIRT7, is associated with lifespan regulation in organisms. SIRT1s play a role in DNA repair and metabolic regulation, and researchers are studying how activating these genes could promote cellular resilience and slow aging. 2. Stem cells in regenerative medicine. Stem cell therapy holds potential for regenerating tissues and organs, which could significantly prolong life and improve the quality of life for the elderly. By reintroducing young, healthy cells into the body, stem cells may help repair damaged tissues, combat degenerative diseases, and reverse aspects of aging. Regenerative medicine also aims to develop lab-grown organs for transplant, which could extend life for individuals with organ failure and reduce the waiting time for transplants. 3. Pharmaceuticals and anti-aging compounds. Rapamycin and metformin. These drugs have shown promise in extending lifespan in animal studies. Rapamycin, for instance, inhibits a protein called TOR, which is involved in cell growth and aging. Metformin, a common diabetes drug, is being researched for its potential to reduce age-related diseases and improve longevity. Senolytic drugs are designed to target and eliminate senescent cells, which accumulate in the body with age and contribute to tissue damage and inflammation. By clearing these cells, senolytics could potentially reduce age-related conditions and improve organ function in the elderly. For caloric restriction and lifestyle approaches, studies have shown that caloric restriction without malnutrition can extend lifespan in animals. This finding has led to research on how reducing calorie intake or intermittent fasting may activate longevity-related pathways in humans, potentially extending healthy years of life. Additionally, lifestyle factors such as exercise, sleep, and stress management have significant impacts on aging. These practices improve cellular health, reduce inflammation, and help delay the onset of age-related diseases. While the complete reversal of aging and death remains an elusive goal, the progress made in life extension research is impressive, offering the potential for longer, healthier lives. Aging research continues to evolve, with ongoing studies into gene therapy, cellular reprogramming, and advanced treatments for age-related diseases shaping the future of human longevity.